I'd like to introduce the next speaker. We have Bo Shi, who is going to talk about PolyA motif prediction. You can start.
Hi, good afternoon, everyone. Welcome to my talk. First, I would like to thank the organizers for providing this opportunity for me to present the work. And this is joint work with Boris, Vladimir, Li, and Xin. Today, I would like to talk about PolyA motif prediction using feature learning techniques. So when DNA is transcribed to messenger RNA, a string of adenine nucleotides is attached to the also known as PolyA tail is attached to RNA. And there's a signaling site consisting of fixed nucleotides upstream in the DNA sequence. And many mutations in the PolyA signal can be associated with this with many diseases, and it's important to detect the PolyA signal. And many existing work have focused on detecting the signal in the RNA. However, it's much more difficult to detect them in the DNA due to the introns and absence of PolyA tail. And existing work focused on using information around sequences in the nearby sequences in the candidate site. Many existing work have focused on devising hand-engineered features from upstream and downstream sequences, and then feed those features into a classifier. However, this requires extensive domain knowledge. Also, it is much harder to come up with new features when performance is already relatively high. So here is an example of some features such as thermodynamics or electron-ion properties. Another line of work focuses on developing very good string kernels. A kernel is a function that measures the similarity between the inputs, and it also corresponds to an inner product between in a high dimension feature space. Many common string kernels usually just count the number of matching substrings in the two sequences. Here in the toy example, we have the feature space, feature mapping corresponding to the kernel just merely counts the number of nucleotides. For example, in the blue sequence, it has two A's. So the feature corresponding to having at least one A, denoted by A colon 1, and having at least two A's, denoted by A colon 2, are both set to 1. It only has one C, so the feature C colon 2, having at least two C's, is set to 0. And this applies similarly to G and T. So in fact, this feature space is infinite, because we may have indefinite number of A's and G's and C and T's. But we can ignore them since their value is 0. And the kernel value is the inner product between these two features, which is 6, which also equals the number of matching nucleotides in these two sequences. Here are a list of common string kernels. The spectral kernel, which counts the number of matching substrings between two sequences, regardless of their locations. In contrast, the weighted degree kernel counts the number of matching subsequences at exactly the same location, which facilitates faster computation, but makes it more sensitive to perturbation and noise. As a balance, the mismatch kernel allows some mismatch, but also matching to same locations. While kernel methods are very powerful, they are very computationally extensive, because you have to compute pairwise distances between all sequences, which scale with the number of sequences squared. In this paper, we propose a different paradigm. Instead of hand engineer those features, we would like to learn them directly from the training set, and then extract the features at test set, and then pass them to the classifier, such as a support vector machine, SVM. Our method is at the intersection of generative models and discriminative methods. Generative models are very good at modeling very complex distributions of the data. But unlike discriminative methods, they do not directly optimize the classification accuracy. Therefore, they often do not get very good results. However, our approach combines those two methods and use generative models to learn and extract features, which is then subsequently passed to discriminative techniques for superior performance. We also employ a fast and efficient learning algorithm through using spectral algorithm. So as mentioned before, generative models can capture very complex structures in the data. In this context, we consider the hidden marker model, HMM, to model the DNA sequences. An HMM consists of a Markov chain of hidden variables, which are not observed. And the observed variables are generated conditioned on the hidden variables. And the HMM provides a very good trade-off between flexibility of the model and also the compressibility. Taking a closer look, the hidden variables actually act as cleaned versions of the observation sequences. And it is this cleaning effect that makes HMMs interesting. In DNAs, the observation sequences are often very noisy, maybe due to mutation or other variations. In terms of classification, the noisiness is exemplified in two aspects. One is diversity, which means for the same location in the sequence nearby the motif, there are many different subsequences that can indicate the class label, whether this is a true motif or false one. The other aspect is uncertainty, which means neither of these subsequences give deterministic information about the class label. An HMM provides a good tool to capture this uncertainty and also clean out the noise. There are some previous attempts in the similar fashion called probabilistic product kernel, PPK. In the PPK, it feeds one HMM to each sequence and then compute pairwise similarities between the HMMs. The drawback with PPK is that you will often encounter severe overfitting due to fitting one HMM to one sequence because of insufficient data. More importantly, in that calculation, the hidden variables are summed out, even though they carry very important discriminative information. At last, the label information is discarded during feature learning. In our method, we only fit two HMMs for the training set, one for all the positive sequences and one for all the negative sequences. Therefore, we are able to learn a more stable model. More importantly, we use the posterior probability of latent variables directly as features and effectively preserving the location-specific information. We also employ a much faster and efficient way to learn the HMMs. Here is the illustration of our model. At the bottom, there's the observation sequence, and the color bars are the posterior distribution of the hidden variables, which are used as features and fed into a classifier. So the disadvantage with the previous scheme is that the conditional independence of the Markov chain induces very short interaction ranges, which means the transition patterns can only be captured up to 2 or 3 nucleotides. We could use a higher order Markov chain to model longer interactions, but that would be much more computationally expensive. A more tractable alternative would be just to combine multiple observation variables into megastates. For example, in this case, ATG is considered a megastate. Therefore, we can significantly increase model complexity with little computational overhead. And here comes the learning part. Traditionally, HMMs are learned by the expectation maximization of the EM algorithm, which is slow and only finds local minimums. In the next few slides, I'm going to present the spectral algorithm that enables faster HMM learning. First, a few basic review about HMM. And HMM is fully defined by 3 parameters: the transition matrix, and the observation matrix, and the prior distribution. Then we define Ft as the posterior probability of the hidden variables at location T, which is also our feature we want to use. And this is recursive update rule for Ft, which involves the transition of the hidden variables and emission of the observation variable xt. We could use an alternative parameterization, which consists of the HTs. The HTs is only different from the original feature vector by an invertible matrix A. Although they're different, they can achieve the same classification accuracy because the invertible matrix A will be canceled out by the weighting vector of the classifier. Why would we want to use the alternative parameterization? If you choose A carefully, we can link the alternative parameterization directly to the observation variables. So we no longer have the hidden variables. We can directly recover the hidden transformed quantities from observation vectors. Here, the C1, C21, C3x1 are the marginal distribution of singletons, pairs, and triples, respectively. They are also called unigrams, bigrams, and trigrams. And U is the leading k left singular vectors of C21, which captures the subspace of hidden variable transitions. As a short summary, our pipeline proceeds first by fitting one HMM for all the positive sequences in the dataset, and one HMM for all the negative sequences. In fitting these HMMs, we only need to calculate the empirical distribution of unigrams, bigrams, and trigrams, and then calculate the HTs accordingly. Once we have the HTs, we can extract features for each sequence. Either they are in the training set or in the test set. And we can concatenate all the features into a long vector and feed them into the support vector machine. And here are some experiment results. On the PolyA detection dataset, it consists of about 15,000 sequences, half positive and half negative, with 12 main variants. Compared with a random forest classifier with 147 hand-engineered features, you can see our method significantly outperforms the random forest model in all cases. And compared with popular string kernels, our method also wins by a large margin. In addition, the training time for our method is significantly lower than the training time of those string kernels because their complexity is n squared. Besides achieving good classification results, our method also provides a convenient way to visualize discriminative regions. We define the discriminative function per location as the conditional expectation of the classifier margin, which means for this subsequence y1 through k, all the sequences that contain it at the location t, we calculate on average what is this classification score. If the subsequence appears mostly in positive sequences, then the score will likely be positive and with large absolute value. If the subsequence mostly appears in negative sequences, then the score will be mostly negative with large absolute value. And if it appears in both types of sequences with almost the same probability, then the score is almost zero, which means it is not discriminative at all. And in practice, the conditional probability can be estimated empirically by sampling from the training data. And here are some visualization results. On the top band is the scores for the dimers. Here, the red color indicates high and positive scores, and the blue color indicates low and negative scores. We can see the dimer AA is a strong positive signal at around up to 30 nucleotides downstream, which happens to be the messenger mRNA cleavage site, which is about 15 to 25 nucleus downstream. On the bottom band, we show the visualizations discriminative score for different lengths of subsequence length from 1 through 5. And because the number of combinations for all the different subsequences is beyond visualization, so beyond dimers, we just sum all the absolute value and show the sum here. So the higher the score, the more discriminative it is. We can see also near 15 to 25 nucleotide downstream, there's a bump here, and it means it is very discriminative. And in summary, we have proposed a new algorithm to automatically extract features from training data, and that bridges the generative modeling and discriminative methods. We have also proposed a novel and efficient learning algorithm for the HMM, and it achieves state-of-the-art results on PolyA prediction tasks. Besides, our method can visualize the discriminative regions of the DNA, providing insight for more analysis. And at last, I would like to emphasize that our technique is not limited to PolyA prediction, but it's a more general framework that can be potentially useful for many other sequence tasks. And at last, I would like to thank you for Akamai Technology for the funding and NSF cost.
Questions? You have to come to the microphone.
Yeah, I like your idea about bridging the gap between generative and discriminative learning very much. But if you extract your features with a generative model, don't you end up with very many features that are similar between the positive and the negative class? And might it be an idea to already learn the HMM discriminatively to find those features for the SVM that separate the classes best?
Actually, we kind of the idea of fitting two HMM instead of one HMM is kind of similar with your idea. So that kind of put some discriminative information in the HMM learning. So instead of fitting one HMM for the whole training set, we are fitting two HMM. So that kind of separate the two sequences.
But both could contain the same features or the same information that is identical between both classes.
Yes, it might, but we have many features, and the feature dimension is quite high. So in the SVM will automatically pick up those features.
Thank you.
Did you compare your method with other programs? Because there are some programs which can detect PolyA motifs and PolyA sites.
We have also tried it on another sequence classification task, which is in the supplementary material. I guess it's about TSS or something.
There are lots of them, like, something like a dozen for human and
what?
several for plants. Different programs. PolyA, Dragon
If
PolyA.
if it's a sequence classification problem, then I think it's maybe.
No, it's for detection of PolyA motifs and detection of PolyA sites. Like SVM PolyA, Dragon PolyA Spotter, lots of programs. So it's your competitors.
What is your question?
So did you compare your method with results of other programs?
Yeah. Oh, not all the problem. I mean, compare it with the cellular result on that data set.
Maybe we can take this offline.
Okay.
So the random forest model you mentioned is actually playing the research published last year on that.
Yes.
That was considered to be one of the state-of-art methods on playing. So we did compare with.
There are like PolyR and SVM PolyA. So it's
Yes,
also.
yeah, exactly. So we compare mainly with the playing.
So there were several additions in several components in the algorithm that she suggested. So it's not entirely clear from the description which one is doing what. So for example, you're saying you're doing the spectral learning for the HMM,
Yes.
right? And then you're on top of it, you're doing the kernel.
Oh, we're
discriminative.
not doing kernel.
You're using the HMM as the features, right?
Yes.
So in principle, for example, if you ran traditional learning of HMM, would that change? Or if you just use the two HMMs and you do a likelihood ratio instead of what you do later, what would happen then? Then it helps to understand what are you doing in terms of what is the relative contribution of each of what you make up as your algorithm. So that's one comment. And the other element is that for the basic training of a generative model, usually generative models you do when you know how the model should look like in terms of the component. If you don't, then you need to do structural learning. But it seems like you predefine the parameters for the HMM, which is generative in nature, in some predefined way, and then it leaves a lot of questions whether that's necessarily the right way to do it and how it affects your learning. Specifically, when you look at the features that you identify, these are the basic signals, but then it leaves the questions of what else you could have found. And because there's a lot of known things about PolyA, it seems a bit could be improved maybe or could be added later, where you would actually use this knowledge to construct your HMM so it would do a better job and then discover new things on top of it.
Yeah, I think we can also include some prior knowledge into the design of the structure of the HMM. Yeah. Thank you.
Thank our speaker again.